1 in 20 segments of some living Australians' genomes traces back to a species science did not even know existed until 2010. Say that number out loud and it sounds like a glitch, like a typo in a lab report. It is not. It is written into the DNA of people alive right now, carried forward from a lineage that vanished tens of thousands of years ago. You came here because of the framing "not human," and it is worth addressing that head-on before we go further. The correction is coming, but first, the mystery. Because the entire physical evidence for this species, the one responsible for that 5%, comes down to a single finger bone. No skull. No skeleton, no face to put to the name. Just one small fragment, pulled from a cave in Siberia, that ended up rewriting what we thought we knew about human origins. Widen the lens on how little that actually is. One bone, one tooth, one jaw, found later, in a completely different mountain range. That is the entire fossil record for a lineage that today lives on in the genomes of millions of people scattered across the globe. Somewhere, in a filing cabinet or a museum drawer, an entire vanished branch of humanity fits inside a box you could hold in one hand. And then there is the paradox that makes this story worth telling. The genetic signal is not strongest anywhere near where the bones were found. It is thickest roughly 6,000 miles away, on the other side of the planet, in populations who have no connection to that Siberian cave beyond deep, ancient ancestry. So what exactly is 5% of you, if it is not modern human, and how does it end up furthest from where it started? The bone did not wait for a body to be found before it was given an identity. Scientists gave it a name before they had a face, before they had a skeleton, before they even had a clear picture of what this population looked like. They called it Denisovan. If this is the kind of thing that keeps you up at 2 a.m. Googling cave excavations, you already know what to do. Public assumption treats the phrase human genome as shorthand for 100% Homo sapiens, no asterisks, no exceptions. That assumption does not survive contact with the data. Nearly every non-African genome on the planet carries a measurable trace of Neanderthal DNA, sitting at around 2%, quietly present in billions of people who have never given it a second thought. But that is only the first layer. A subset of humanity carries a second archaic signal, larger than the Neanderthal trace, and it belongs to a completely different lineage. Before we get to the number attached to it, it is worth naming exactly who carries it. Aboriginal Australians, Papuans, certain groups across the Philippines. These are the populations where this second signal shows up most strongly, concentrated in genomes that trace back to some of the oldest continuous human lineages on the planet. This is not contamination. It is not a sequencing error, not a fluke in the lab, not noise in the data. It is inheritance, the direct genetic residue of interbreeding events that took place tens of thousands of years ago between modern humans and a population that no longer exists in any recognizable form. What sits in these genomes today is a record of contact between two branches of humanity that diverged, met, and then one of them disappeared. That inheritance carries weight beyond curiosity. It makes the genomes of these specific populations some of the most scientifically valuable material on Earth, for reconstructing lineages that left almost nothing else behind. Where the fossil record goes silent, these genomes speak. They are, in a very literal sense, walking archives of an extinct branch of the human family tree, carrying information that no bone, no excavation, no museum collection has been able to supply on its own. To understand how anyone even learned to read that archive, the story has to go back to the discovery that made it possible to measure any of this in the first place, in 2010, in a cave in southern Siberia. Denisova Cave sits in the Altai Mountains of southern Siberia, a stretch of remote, weathered terrain that had already been excavated for years before it produced anything that would upend the field of human genetics. What it produced was small enough to overlook: a single finger bone. In 2010, Reich and colleagues published their findings in Nature after extracting DNA from that bone and sequencing it to roughly 1.9x genome coverage. Thin by modern standards, but enough to reveal something no one was expecting. The DNA did not match Neanderthal. It did not match modern human. It belonged to something else entirely. The individual behind that bone was a young female, later dated to roughly 50,000 years old or more, from a population that shared a common origin with Neanderthals before splitting off on its own separate path. And the finding that mattered most, stated with precision rather than speculation, was this: This population contributed between 4 and 6% of its genetic material to present-day Melanesian genomes. The finger bone was not alone in that cave layer. A molar tooth, recovered from the same stratum, carried mitochondrial DNA nearly identical to the bones. A second individual, corroborating the first, confirming this was not a fluke of one fragment but the genetic signature of an entire population. The tooth itself was strange to look at. Its morphology did not match Neanderthal teeth, and it did not match modern human teeth either. It was physically alien on the examination table, and yet genetically, unmistakably present in the bodies of living people today. With no jaw, no cranium, no way to reconstruct a face or a name from history, the research team made a naming choice rooted entirely in geography. They called this population Denisovans, after the cave, because there simply was not enough skeletal material to justify calling them anything more descriptive. From that cave in Siberia, the trail leads directly to the place where this percentage turned out to run highest of all, Aboriginal Australia. Set the two lineages side by side and the imbalance is immediate. Neanderthals left behind hundreds of skeletons, scattered across sites in Europe and the Middle East. Enough material to reconstruct posture, build, even elements of behavior. Denisovans left a finger bone, a molar, and, as later discoveries would show, a jawbone found far from Siberia. That imbalance cuts against intuition once you look at the genetic record. In some living populations, the Denisovan genetic footprint is actually larger than the Neanderthal one, despite this near-total fossil silence. A species can vanish almost completely from the physical record and still leave a louder signal in the bodies of the people descended from it. The next piece of physical evidence did not surface in Siberia at all. In 2019, a jawbone was identified from Baishia Karst Cave in Tibet, confirmed as Denisovan and geographically distant from the original cave where the story began. It remains, alongside the finger bone and the molar, one of the only confirmed physical Denisovan fossil finds anywhere on Earth. With so little bone to work from, scientists had to build their picture of this population using an entirely different method. Instead of skeletons, they compare whole genomes, living people and ancient remains alike, piecing together an entire vanished population out of statistical shadows left behind in DNA. It is reconstruction without a body, a population rebuilt from inheritance rather than excavation. And it is worth correcting the framing directly here. Denisovans were humans, a distinct, now extinct branch of the human family, closely related to Neanderthals and to us, not some separate non-human species in any colloquial sense of the word. The next chapter of the story required a genome that did not exist yet. The story shifts continents in 1923, aboard a train traveling from Sydney to Perth. British anthropologist Alfred Court Haddon was on that journey, and somewhere along the route, a young Aboriginal man from the Goldfields region of Western Australia offered him a lock of hair. That hair was catalogued, stored, and then largely forgotten as anything more than a historical curiosity. For roughly 90 years, it sat untouched, its genomic value unrecognized, waiting in a collection while the science that could make sense of it had not yet been invented. It was S.K. Willerslev's team at the University of Copenhagen that recognized what that lock of hair actually represented: a way to obtain an uncontaminated, pre-colonial Aboriginal genome, free from the European ancestry mixing present in most living samples collected after colonization. A single strand of hair, a century old, offered a genetic snapshot that no living sample could match. The process that followed was not treated as a simple lab procedure. Before sequencing began, the study was reviewed and approved by both a Danish bioethics board and an Aboriginal community council, a dual layer of oversight built into the research from the start. The findings were published as Rasmussen et al. 2011 in Science, Volume 334, pages 94 through 98. The hair itself was only a century old. What it revealed reached back 60,000 years further than that. The genomic comparison ran against reference genomes from Han Chinese, European, and African populations, and the result was unambiguous. Aboriginal Australian ancestors branched off from the Eurasian lineage somewhere between 62,000 and 75,000 years ago. That number matters because of what it predates. It comes tens of thousands of years before the split between the ancestors of modern Europeans and the ancestors of modern Asians. In other words, this branching happened before Europe and Asia even had genetically distinct populations to speak of. The implication reaches back to the very beginning of the human journey out of Africa. Aboriginal Australians descend from one of the very first waves of people to leave the continent, push deep into Asia, and continue onward until they reach Sahul, the ancient landmass that connected Australia and New Guinea. That places Aboriginal Australian populations among the oldest continuous populations living outside Africa anywhere on Earth today, a living link to the earliest chapter of human migration. But the model built on that lock of hair did not stand unchallenged. This separate early dispersal theory was proposed, published, and immediately contested. It was not universally accepted, not folded quietly into consensus, not treated as settled the moment it appeared in print. Not every geneticist agreed. That same year, 2011, a study led by David Reich took a different approach entirely, analyzing 300 genomes drawn from 142 populations spread across the globe. The conclusion this team reached cut directly against the separate dispersal model. Their analysis argued that indigenous Australians, New Guineans, and Andamanese populations do not derive substantial ancestry from a distinct early migration out of Africa. Instead, they share the same out-of-Africa source as every other non-African population on the planet. This is not a settled question dressed up as drama for the sake of narrative tension. It is a real, still-referenced methodological disagreement within the field, centered on how many times humans left Africa and when each of those departures actually happened. Researchers on both sides of this debate are working from real data, real models, and real uncertainty about how to interpret deep population history. What both camps agree on, regardless of which timeline turns out to be correct, is this. Aboriginal Australian and Papuan ancestors picked up something that no European lineage and no East Asian lineage acquired in comparable quantity: Denisovan DNA. The disagreement over exactly when the ancestral split happened does not touch that finding at all. The archaic admixture itself has been confirmed independently, repeatedly, across multiple studies and multiple methods. 5 years later, a much larger dataset arrived to test both models against real numbers. Malaspina and colleagues published their findings in 2016, in Nature, volume 538, pages 207 through 214. And the scale of this study dwarfed everything that came before it. 83 high-coverage genomes from Aboriginal Australians, drawn from 8 distinct Pama-Nyungan language groups, alongside 25 Papuan genomes from the New Guinea highlands. To understand why that number mattered, consider what had existed before it. Every piece of demographic inference about Aboriginal Australian population history had rested on just 3 prior genomes. One came from that single lock of hair. 2 others came from cell lines whose provenance was uncertain at best. An entire continent's deep genetic history, reconstructed from a sample size that could fit on one hand. The lead researcher, Anna Sapfo Malaspinas, working through the Swiss Institute of Bioinformatics and the University of Bern, did not run this study in isolation. Aboriginal community co-authors were directly involved in sampling and consent, embedded in the research process rather than treated as subjects of it. What the expanded dataset revealed reshaped assumptions about internal diversity within a single continent. Southwestern desert Australian groups turned out to be more genetically different from northeastern Australian groups than Native Americans are from Siberians. 2 populations, separated by an ocean and a continent in one comparison, showed less genetic distance than 2 groups living within the borders of Australia itself. That internal diversity turned out to have a date attached to it. Papuan and Aboriginal Australian ancestors diversified from each other somewhere between 25,000 and 40,000 years ago, evidence that population structure inside Sahul was already forming well before the current Holocene period even began. This was not a single unified group spreading uniformly. It was a landmass already fracturing into distinct populations tens of thousands of years before recorded history. Despite that internal divergence, the genomic data traced every sampled Aboriginal Australian back to a single founding population, one that itself differentiated further somewhere between 10,000 and 32,000 years ago. A shared root, followed by branching, followed by more branching still. The study also detected a distinct population expansion moving out of northeast Australia during the Holocene, within the last 10,000 years, accompanied by limited gene flow outward from that expansion. That pattern lines up closely with the archaeological spread of Pama-Nyungan languages across the continent, genetics and linguistics pointing toward the same underlying movement of people. Beyond migration and diversification, the researchers flagged selection signatures in the genome, potential evidence of adaptation tied specifically to survival in Australia's desert interior. The combined estimate places the split between Aboriginal Australian and Papuan ancestors and the broader Eurasian lineage somewhere between 51,000 and 100,000 years ago, depending on which demographic model is applied. None of that explains where the Denisovan DNA came from. That required looking at the percentage itself. Across the body of research, the stated range holds steady. 4 to 6% Denisovan ancestry in Aboriginal Australians and Papuans, anchored by the original Reich et al. 2010 baseline, with some regional estimates landing closer to 2 to 3%. Set that beside the Neanderthal figure for comparison. Neanderthal ancestry sits at around 2% in nearly all non-African populations worldwide, a number that stays remarkably consistent no matter where on the globe you sample. Denisovan ancestry behaves nothing like that. It is wildly uneven across populations, sitting at near zero in Europeans, staying low across mainland East Asia, and climbing to its highest recorded levels specifically in Sahul, meaning Australia and New Guinea, along with parts of island Southeast Asia. That unevenness is not a footnote. It is itself the data point that tells geneticists something specific happened in a specific place. Interbreeding with Denisovans did not occur everywhere modern humans happened to pass through. It occurred somewhere in particular, concentrated enough to leave this uneven signature behind. The obvious candidate for that location is the Sunda region of Southeast Asia, the logical geographic bridge between Siberia and Sahul. And yet that region has never produced a single Denisovan fossil. Not one bone, not one tooth. Nothing to physically confirm what the DNA keeps pointing toward. The bones say Siberia and Tibet. The DNA says somewhere else entirely. Here is a population that geneticists are confident lived, thrived, and interbred in Southeast Asia and the region known as Wallacea, and yet not a single physical trace of them has ever been found there. No bone, no tooth, no fragment of any kind. Alan Cooper, from the University of Adelaide, made a pointed observation about this publicly. He noted that an admixture level sitting around 2 to 3% suggests something specific about the timing and distance of this interbreeding. It doesn't read like a diluted signal carried forward from a deep, distant Siberian ancestry. It reads like something closer, something more recent, a nearby encounter rather than a faded genetic echo from thousands of kilometers away. That places this missing population squarely in the category geneticists call a ghost lineage, a population known only through the DNA carried by their descendants, never once confirmed through their own physical remains. They exist in genome after genome, and nowhere in the ground. Consider the scale of that mismatch. 2 confirmed Denisovan fossil sites exist on the entire planet, one in Siberia, one in Tibet. Against that, the genetic footprint runs strongest thousands of kilometers to the south and east, in populations with no proximity to either site. That gap raises an open question the field has not resolved. Either paleoanthropologists have simply been searching in the wrong places, missing sites that haven't yet been excavated, or tropical Southeast Asia's climate destroyed the bone before it ever had the chance to fossilize, erasing the physical evidence while leaving the genetic evidence intact. In 2019, a new study stopped treating this as one interbreeding event and started treating it as several. Jacobs and colleagues published their findings in 2019 in the journal Cell, volume 177, pages 1010 through 1021. Their study sequenced 161 new genomes, drawn from 14 island groups spanning island Southeast Asia and New Guinea, one of the broadest genomic surveys of the region ever assembled. What they found reshaped the entire picture of Denisovan interbreeding. The DNA blocks showing up in modern Papuan genomes did not trace back to a single Denisovan source population. Instead, they clustered statistically into 2 distinct groups, labeled D1 and D2, each with its own genetic signature. The divergence between D1 and D2 was not a small gap. These 2 groups split from each other more than 350,000 years ago, a longer separation than the split between modern humans and Neanderthals under some estimates. 2 branches of the same broader lineage, already distinct from one another for longer than our own species has been distinct from Neanderthals. The implication reaches further than a simple subdivision. Denisovans were never one unified population spread across Asia. They were multiple, geographically isolated groups, and modern humans interbred with more than one of them, in separate encounters, at separate times. The geographic footprint of these 2 groups differs sharply. T1 shows up almost exclusively in Papuan genomes, concentrated and localized. D2, by contrast, carries a far wider footprint, turning up across a broader stretch of Asia and Oceania. The statistical backbone of this finding held up under scrutiny. A Gaussian mixture model applied to the data strongly favored two distinct peaks over a single unified one, giving the two population picture real mathematical weight rather than speculation. Move east of the Wallace Line, and the story of D2 changes again. The Wallace Line marks a biogeographic boundary running through the Indonesian archipelago, separating the fauna characteristic of Asia from the fauna characteristic of Australia. It is one of the sharpest ecological divides on the planet, and it plays directly into this story. Jacobs and colleagues found evidence that the D2 lineage's genetic introgression continued even after modern humans had already crossed that line, moving into Wallacea and onward into Sahul. This was not a single mainland Asian mixing event whose signal simply grew fainter as people spread outward. Interbreeding kept happening as modern humans moved through the island chains of Southeast Asia itself. That finding carries a logistical weight that reshapes assumptions about Denisovan capability. For interbreeding to continue past the Wallace Line, Denisovans would have needed to cross open water or navigate an island chain themselves, a level of mobility not typically associated with archaic hominin populations in older models of human dispersal. A third signal complicates the picture further. Researchers identified a less divergent lineage labeled D0, appearing mainly in East Asians, Siberians, and indigenous Americans. This signal closely matches the reference Altai genome, the same genetic material recovered from that original finger bone in Siberia, pointing to yet another separate mixing event, this one more northern in origin. A second team, working independently, reached the same conclusion from entirely different data. Browning and colleagues published their own findings in 2018, in Cell, Volume 173, pages 53 through 61, under the title, Analysis of Human Sequence Data Reveals Two Pulses of Archaic Denisovan Admixture. This team worked from separate genomic datasets, using different methods than Jacobs and colleagues, and arrived independently at the same core conclusion. There were two distinct pulses of Denisovan admixture into the ancestors of Oceanian populations, not one. Two independent research groups, using different statistical approaches and different source data, converging on the same multi-event picture. In population genomics, that kind of independent convergence carries real weight. It is not one team's interpretation standing alone. It is confirmation. What this rules out matters as much as what it confirms. The old textbook model, one interbreeding event somewhere in Asia followed by simple human dispersal outward, no longer fits the data at all. That model has been replaced by something far more layered. Reconsider the 4 to 6% figure established earlier. That number was never a single clean inheritance from one ancestral encounter. It is the residue of several separate encounters, layered on top of one another, occurring in different places, at different points in time. The clearest evidence for how extreme this layering can get isn't in Australia at all. It's in the Philippines. Larena and colleagues published their findings in Current Biology in 2021, drawing on roughly 2.3 million genotypes analyzed across 118 ethnic groups throughout the Philippines, including 25 self-identified Negrito populations. Their central finding reset the record books. The Ayta Magbukon people carry the highest documented Denisovan ancestry recorded anywhere on Earth. That figure runs 30 to 40% higher than the levels found in Aboriginal Australians and Papuans, the very populations previously assumed to hold the world record for archaic ancestry. The study's authors concluded this was not simply an extension of the same Sahul mixing story already established. It represents a separate, independent admixture event entirely, its own distinct chapter in the broader Denisovan story. A single data point from a study like this is enough to rewrite the ranking that had stood since 2010, when the original Siberian fingerbone first put a number on Denisovan ancestry in living people. Researchers went further still, explicitly linking this finding to Homo luzonensis, a separate archaic hominin species identified from fossils found in the Philippines. That connection raises the possibility of a genetically related population, distinct and largely unaccounted for, still waiting to be understood. One island chain, at least 2, maybe 3, extinct human lineages, tangled together. Homo luzonensis is a separate archaic hominin species, identified from fossils recovered on Luzon Island in the Philippines, and formally classified as distinct from both Denisovans and Neanderthals. Larena and colleagues raised a possibility in their study without confirming it outright. Multiple archaic species may have inhabited the Philippines before modern humans ever arrived there, and some of these lineages could be genetically related to one another, part of a broader web of archaic populations spread across the islands. No genome has ever been recovered from Homo luzonensis fossils. That absence matters. Without genetic material to compare directly, the connection between this species and the Denisovan signal found in living Filipino populations remains inference. It is a plausible hypothesis, not proof. That gap leaves a real, unresolved question sitting at the center of the field. Is the record-high Denisovan signal found in the Ayta Magbukon actually pure Denisovan ancestry, or is part of it something else entirely, currently misclassified simply because there is no comparison genome available to sort one archaic lineage from another? This deserves to be framed honestly, as an active gap in ongoing research rather than a mystery already solved. It is precisely the kind of open question that a single new fossil, or a single ancient genome pulled from the right site, could overturn completely. None of this explains what the DNA is actually doing inside a living person today. That part of the story starts in the immune system. The story shifts here, moving away from ancestry and deep history and toward function. The question changes from where this DNA came from to where it sits in the modern genome, and what it actually affects in the people who carry it. Multiple independent studies converge on the same answer. Immune-related genes make up the dominant category where Denisovan DNA concentrates. TNF-AIP3, OAS2, OAS3, along with PELI2, BANK1, CCR10, CD33, DDX60, IRF4, and JAK1, a cluster of genes tied directly to how the human immune system functions. A genome scan posted on bioRxiv applied a two-phase detection method across 20 Papua New Guinean and Australian genomes, and the scale of what it found was substantial. 1.34 gigabases of Denisovan introgressed sequence, containing 38,877 individual Denisovan-derived alleles scattered throughout the genome. Within that enormous set, 78 regions showed clear signs of positive selection, meaning evolution actively favored these specific segments over generations rather than simply carrying them along as passive genetic baggage. And 121 of the introgressed alleles are missense variants, meaning they actually change the protein that gets produced, not just sitting quietly in background sequence with no functional consequence. The significance of that distinction is direct. This is not inert, archival DNA sitting untouched as a marker of ancient ancestry. Some of it is doing active biological work in living people, right now, in this generation. One gene in particular has now been tested directly in the lab, not just inferred from statistics. Zammit and colleagues published their findings in 2019, in Nature Immunology, Volume 20, pages 1299 through 1310, and their work moved this story out of pure statistics and into direct experimentation. The gene at the center of their study is TNF-AIP3, which encodes a protein called A20. That protein functions as a molecular brake on two major inflammatory signaling pathways in the body, NF-kappa B and TNF, both central to how the immune system regulates inflammation. The research team took Denisovan, modern human, and mouse versions of this gene's alleles and compared them directly, side by side, in controlled laboratory conditions rather than relying solely on genomic inference. They identified a specific Denisovan haplotype, T108A, I207L, a distinct genetic signature carried within this immune-regulating gene. What they found was that this Denisovan variant measurably tunes A20 phosphorylation. In plain terms, it changes, in a testable lab assay, how strongly the immune system's inflammatory brake actually engages when activated. This is not a correlation pulled from population data. It is a direct, observable effect, measured in the lab. That distinction moves the entire story from statistical association to direct experimental confirmation of function. This piece of inherited Denisovan DNA does something specific, something measurable, inside the modern immune system. A brake that tunes differently is not automatically a better brake. A study published in PLOS Genetics in 2022 used a massively parallel reporter assay tested across 56 Papuan genomes to examine Denisovan variants near TNF-AIP3, OAS2, and OAS3 directly, pushing the research even further into functional territory. The results were specific. Researchers identified 20 Denisovan variants tied to the regulation of OAS2 and OAS3. Of all the Denisovan sequences tested in the assay, 8.22% showed active gene regulatory effects. meaning they were doing measurable work in controlling gene activity. And roughly 9.22% showed a measurably different level of activity when comparing the Denisovan version of a given site against the modern human version of that same site. The researchers' interpretation of the TNF-AIP3 effect specifically carries real nuance. Increased A20 activity could reduce chronic inflammation and lower autoimmune risk, but that same increased activity potentially comes at a cost: a slower or more blunted immune response when the body actually needs to fight off infection. This is presented in the scientific literature as a genuine, open trade-off. Not a settled story about a good gene or a bad gene, the kind of clean narrative that tends to survive in headlines, while the actual nuance gets lost along the way. Tie this back to the OAS gene cluster specifically. Its role centers on antiviral defense, a separate infection response pathway entirely from the TNF-AIP3 inflammation story, and yet both pathways were independently shaped by Denisovan inheritance, operating through completely different biological mechanisms. This is precisely why researchers increasingly treat that roughly 5% figure as biologically active, present-tense inheritance. Not archival trivia sitting quietly in the background of a genome, but something with ongoing consequence for the people who carry it. None of this required Denisovans to be anything other than human. It required them to be a different kind of us. Every Aboriginal Australian and Papuan genome sequenced to date confirms the same pattern independently. This is not a one-off anomaly, not a fluke result from a single study that failed to replicate. It holds study after study, population after population. Several questions remain genuinely unresolved. The geography is unresolved. No Denisovan fossil has ever been recovered from Southeast Asia, from Wallacea, or from Sahul, despite the strongest genetic signal on the entire planet sitting precisely in that region. The taxonomy is unresolved. Whether the Philippine Denisovan-like signal connects to Homo luzonensis remains unproven, an open hypothesis waiting on evidence that does not yet exist. And the function is unresolved. Researchers are still working through which of the thousands of introgressed alleles actually matter, and which are simply carried along for the ride, functionally inert. Running underneath every study cited across this entire investigation is the same ethical through-line. From the 1923 hair sample on a train to Perth, through to the 2016 Genome Project drawing on 83 Aboriginal genomes, community consultation and consent shaped what was studied and how it was studied at every stage. Return, one final time, to where this all began. The framing deserves full correction, stated plainly. This is not a lesser lineage. It is not human, despite how that phrase was used to draw you in at the very start. It is a separate, extinct branch of the human family, a branch whose only remaining trace, across most of the world, is a bone and a tooth, and whose presence, in a few living populations today, runs as high as 1/20th of an entire genome. If a whole species can vanish and leave nothing behind but a fraction of a percent in someone's blood, what else is still hiding in the rest of the code?